Matt Might is an associate professor of computer science and a presidential scholar at University of Utah, and he is a true dyed-in-the-wool computer scientist working from everything from compiler design to cybersecurity, and yet he seems to have published recently some articles in the biomedical literature, so go figure. Matt, thank you very much for coming.
One second, well, there we go. All right. Well, thanks for having me, Zach. It's really a pleasure to be here. So I'm here to tell you a story. This is a story about precision medicine, and in fact is the story of my son. This is the story of what happens when you're told, as we were 3 years ago, that your child is the first and only patient ever discovered with their particular, now new, ultra-rare genetic disorder. So in the aftermath of this diagnosis, we had to start answering questions. And at the top of that list was, what do you do when you have an N of 1 diagnosis? What do you do when you're the first patient in the world? And so for the past 3 years, we've been mapping out what I'm tentatively calling sort of the precision medicine algorithm, just sort of feeling our way along for what you do when this happens. What I hope to do in this talk is really take you through this pipeline from undiagnosed at the beginning, through some magic process in the middle, to being healthy out on the other side. So this talk is really about trying to sketch out how we might demystify that middle part of this algorithm. And in case you want to go to sleep or you don't want to pay attention, I can summarize it for you in 3 words, at least the initial phases of this algorithm, which are, try the internet. And this is great. Because the internet is cheap, and unless you happen to live in, say, North Korea, it is readily available. So, yeah, pretty much everybody can get access to Google, and it's amazing. I'll show you what you can do with access to Google these days. So even though this is the story of my son Bertrand, this is a story that in many ways is being told thousands of times over at this point. And so even beyond rare disease, I mean, I think, as was already mentioned, cancer is also something you can view through the lens of precision medicine. And so this algorithm doesn't apply to just rare disease. This is really sort of any genetic disease in general. All right, so for an algorithm, you need an input. And the input to the algorithm in this case is patients, undiagnosed patients. So I'm going to show you our sort of the undiagnosed phase of our journey very quickly. There's a phrase a lot of patients use for this part, which they call being trapped in an undiagnosed island, where it's an island with a population of one or one family. And when you're trapped on this island, you don't know why your child is suffering. You don't know why your child, like in our case, has seizures, or why they have an extreme developmental delay, or why they have this intractable movement disorder, or curiously enough, in the case of my son, why he couldn't cry liquid tears. So it's a terrible place to be trapped on an undiagnosed island. But it's also an island where you learn about that strength in the human spirit and the quiet power of resilience, of never giving up and never giving in. So that was our life for 48 months. We were trapped on that island. And then we found step 1 of the precision medicine algorithm, which is sequencing. So this is back in 2010 when we tried to get sequencing initially, because we had tried every means available for a traditional diagnosis, and obviously we could have kept going on that route as long as we wanted. We were never going to get our answer, but we'd exhausted every reasonable diagnosis when he was about 3 years old. Now back in 2010, exome sequencing was pretty new. You couldn't get it in a clinical setting, so we were very lucky that Duke University stepped up and said, we will do it in a research capacity. We will do exome sequencing as part of a research study. And so what they did is they sequenced the 3 of us. They sequenced me, they sequenced my wife, and they sequenced my son. And they checked for de novo variants, but also interesting variants that he'd potentially inherited from both of us. And what they found was he had inherited 2 loss-of-function alleles, one from me, one from my wife, obviously, in his NGLY1 or NGLY1 gene. And then when they told us about this, they also were careful to clarify that he was the first, and in fact the only patient that had ever been seen with a loss of function in this gene. And as a result, it was sort of like, well, we think this is it. And so that put us onto what I would call step 2 of the algorithm, which is trying to make sense of the results that come back from sequencing. Because you get a lot of variants, and we actually had other variants in this one too, but this is the one deemed most promising. So we had to do some, well, Duke really had to do some homework. So what they did is they checked to make sure, is this really knocking out the protein? And sure enough, this really was knocking out the protein. There wasn't even, not even the mutant protein was surviving. So that's a promising step. And then what they did is they took my son and interpreted his phenotype as a set of data points. And they checked, can we fit this phenotype back to the hypothesized genotype in this case? And so to do that, they had to say, well, what is this genotype telling us? What is it telling us to have a loss of function in this particular gene? So we just sort of, at this point, they started examining what this gene does. They said, well, this gene is involved in a pathway that deglycosylates misfolded glycoproteins. So we can imagine that if this protein were missing, then if this pathway were gone, what we'd have is an accumulation of misfolded glycoproteins in patient cells. This is an observation that you could go start to check. And to check this, they turned to an expert in glycobiology. They turned to Dr. Hudson Freeze at Sanford Burnham. They said, you're an expert in disorders of glycosylation in human beings. And if we told you we had a patient that was missing this particular enzyme, what do you think you'd see? And the picture he drew was my son. So at this point, with no other patients out there, we said, well, this is about as certain as we can get that we've got it. Because this looks like a match between genotype and phenotype here. But they're also very clear that in the event of an N of 1 diagnosis, it's not actionable. There's nothing you can do in a traditional medical sense when you're the only patient in the world. I mean, there's obviously no drug waiting for you when this happens. Which leads me to step 3A in the algorithm, which is to do some science. Because in the absence of any available medication, it's science itself that becomes the medicine. And science itself becomes action. So I think, if there's one thing I could do as a patient, and if I could implore all of you here to do, it would be to eliminate the phrase not actionable from your medical vocabulary. Because in this day and age, it simply doesn't make sense anymore. Because once you finally get a diagnosis, you go from maybe dozens, potentially even hundreds of possible targets that you're shooting at, down to one overnight. Once you have one target, you can do good science, and that's what we started doing. Once you have a target, you start looking for arrows. So we started looking for arrows as soon as we got this answer. And so the first thing we did is we checked PubMed. We said, okay, well, PubMed, tell us about NGLY1. Trying to stand on the shoulders of giants here, today you'll get 11 papers on NGLY1, which is about double what you had 3 years ago. So one thing we realized very quickly is we're going to need more giants, because there was not a lot known about this particular enzyme. So within a few weeks, we were actually out at Sanford Burnham to meet Dr. Freeze, and we began a collaboration that's actually continued to this very day. And what we did is we said, okay, well, first, let's just show us how far we've come. Can you just give us a sense of the landscape in glycobiology and the role of this enzyme? And what he did was he took us to the peak of our diagnostic odyssey. And from here, we could finally see the wilderness through which we'd been wandering. But then he showed us how far we would have to go to reach understanding, treatment, and cure, which, as we quickly learned, is significantly more challenging than just getting an answer. And I think this is actually a reasonable metaphor for the current state of precision medicine. It's like everybody gets their diagnosis, and it's like, all right, go climb Everest now. That's pretty much what we do. And obviously, this is going to change, but it's going to change because of the research that's happening today. But this is really the state of affairs when you get an N of 1 diagnosis. But the amazing thing is that very shortly after getting this diagnosis, we were actually able to start taking some concrete action. So working with Dr. Freeze, we said, well, one of the mutations is a premature stop mutation. So maybe we can focus on some therapeutics for that angle. So if you're not familiar with premature stops or read-throughs, here's the idea. The idea is that one of the alleles, the one he got from me, in fact, was just truncating the construction of the protein prematurely. And there are compounds out there called read-through compounds, which can actually potentially allow full translation of the protein. So we thought, well, let's try some of these. So we tried a whole bunch of them on fibroblasts in a dish, and unfortunately, none of them worked. Now, the thing is, some people would think that this might be disheartening, but it was actually really encouraging to see this, to work through this initial process, because this was all like weeks after getting the diagnosis, maybe a couple of months before we started getting some early results back on these read-through compounds, because we went from being lost in the wilderness, stuck on that island, having no idea what he had, to suddenly taking plausible attacks on the disorder just weeks later. I mean, it was tremendous, I think, to go from that wilderness to actually the laboratory so quickly. And we realized from this that there's no way we could do this alone, that this is going to be a very long journey. And in addition to working with scientists, we really had to find other patients too. One patient versus the world was going to be a very difficult battle. Which brings me to step 3B in the algorithm, which is finding a community. So I think there's a lot you can do today in this regard. And one of the first things we did was we actually looked at an exome database. So at the time, it was the Washington database. And from these databases like the Washington database and the EXAC database, you can get an estimate of the number of patients out there by looking at the frequency of the alleles you're interested in. And when we did those calculations, we came up with maybe 500 or so living patients, depending on how you estimate mortality. And so we thought, well, you can do a lot with 500 if we could start to find them. So we said, let's go find other patients. Let's see where they are. So to do that, I wrote a blog post. And I very carefully designed this blog post to do a couple things. The first thing this blog post needed to do was it had to go viral, because I needed to get this thing out in front of as many eyeballs as I possibly could, so that it would hopefully intersect with other patients or physicians caring for other patients, and then bring them back to us. The other thing I wanted to do was to rank as highly as possible in Google for the search terms of interest to patients and to parents. So that if patients and parents were out there Googling their own symptoms or things related to their disorder, they would have a good chance of landing on this page. So we essentially employed search engine optimization to help find other patients. So I'll show you the blog post I wrote. It was 5,000 words long, and this is the top part right here. And I think you can see from the picture that I did engineer this thing to go as viral and spread as quickly as possible. And the amazing thing is it actually did. This thing took off like wildfire, and before I knew it, it was all over social media. So places like Reddit, it was reposted in Gizmodo, it was the top of Hacker News. So this got out and it got seen by a lot of people, and the right people, very, very quickly. And as a matter of fact, it started snowballing from there, because even traditional media started taking an interest. And in fact, it ended up in the New Yorker last summer. In fact, the author of this piece, Seth Mnookin, is here today. And this put it out there in a whole different way and put us on an even more interesting trajectory, some of which I'll talk about in just a moment. But the fact of the matter is it also did start to rank very highly, so that if patients were out there Googling for the key terms related to this disorder, if they were holding a diagnostic slip that said NGLY1, and they typed it into Google, they were going to find us now. Or if they were typing in some of the more obscure terms, they were going to find us. And the amazing thing is, they did. So within a couple months, 2 patients actually showed up in Turkey, 2 siblings. And a few months after that, we had 2 patients in Israel, and again, 2 more siblings. And then, about a week or 2 after that, we had the second US patient in Palo Alto. And finding that second US patient, making contact here in the US, I think was really the genesis of the NGLY1 community. Once we found another US patient, things really started to move very quickly in terms of finding other patients, but also really moving towards understanding treatment and cure. And the amazing thing is that over the past 3 years, patients have been popping up all over the world. So it's not just here in the US, it's not just, it's I think every continent except Australia and Antarctica at this point. And what this has really done, to have so many patients in such a short amount of time, is it's really washed away that dark night of uncertainty and replaced it with the light of science and community. And it's a community which, as you'll find across rare disease, is incredibly united. We're all working together behind understanding, treating, and curing this disorder. And community today stands at about 37. And the amazing thing is that every time I speak on something like this, I actually have to update the numbers. So we found 2 more patients just last week. And the rate at which we're finding patients is actually accelerating. So I think this is due to both the combination of us getting even better at getting into the search results, but also the fact that sequencing is becoming so widely deployed. And now we can ask questions like, what can you do with an N of 37? And it turns out there's a lot you can do once your patient population starts to grow. So I think one of the things that happens right off the bat is you start to get natural experiments. And for those of you following along, this is step 3B.A in the algorithm. I'll stop labeling them the numbers. I'm going to give you a flow chart towards the end. Yes. I'll give you the flow chart with everything at the end. And I want to share, there's actually been, I think, 3 or 4 really interesting patients that, in and of just by existing, have told us something about this disorder, something potentially actionable about this disorder. I'll share one of those stories with you. So this child right here, who I met about 6 months ago, not only has NGLI1 deficiency, he also unfortunately had aggressive pediatric liver cancer by the age of 2, necessitating a full liver transplant. And the interesting thing about this patient is that after his liver transplant, his seizures actually went away entirely. And not just his seizure, but his EEG completely normalized. These patients do not have normal EEGs. This child now has a normal EEG. So now we have to wonder, is a liver transplant a reasonable therapy for all the patients? And is it really the new liver, or is it potentially the heavy immunosuppressants he's on? The fact of the matter is that patients like this give the scientists routes to explore. And unearth questions we need to have answered, and ultimately give hope to the patients. And there's multiple patients like this in the community now that are different for some reason, and by studying those differences, we're learning a lot about the disorder. When you have a patient community, you can also have conferences. So I think about 1.5 years after the disorder was discovered, we actually had the first patient science parent symposium out at Sanford Burnham, where I think it was the time there was about 8 patients that we were all able to get together and meet for the first time and to work with the scientists and say, well, what's the right direction to go here? How should we use our limited resources to push this disorder forward? When you have communities, you can also create foundations. So we have not 1, but in fact 2 patient-led foundations today for understanding, treating, and curing this disorder. That's a ratio of foundations to patients that if we can maintain going forward, I think bodes quite well. When you have enough patients, you can also do natural histories with a level of precision to get reasonable statistical confidence, reasonable statistical significance in the results that come out. We've been very, very lucky that at about the same time this disorder was being discovered, the NIH was also setting up a natural history study for congenital disorders of glycosylation, and we happen to be a brand new congenital disorder of glycosylation. So we got NGLI1 eligible to participate in the protocol. And in the past year, about half of all known patients have moved through this and been deeply phenotyped in the process. So I'm very grateful to folks like Lynn Wolf, who's been leading up the research at NIH, and Bill Gall for opening the protocol to us. And just to give you a sense of what deep phenotyping looks like, because I think deep phenotyping is actually a really important part of precision medicine going forward. What I have is a very quick slideshow of 1 picture for every poke, prod, procedure, or appointment that occurs over the course of this week-long admission to NIH. And this is something that these kids do every year. So my son did this for the first time a year ago. He just went back 2 weeks ago. And it is incredible, the data coming out of this, when you have close to 50% of all known patients phenotyped this deeply. So when you do this, what you get out of it, well, if you're lucky, and we have been lucky, are biomarkers, clear clinical indicators of what's going on with this disorder, and things that can potentially serve as endpoints if we move towards clinical trials with some of the compounds that we're considering. And so just for example, like what they found with these patients is that they have abnormally low protein in their spinal fluid, which is strange. Usually with a neurodegenerative disorder, you'd expect to see higher protein. For some reason, these kids have lower protein. And also somewhat, interestingly enough, in a disorder in which we have an error of deglycosylation, we're actually seeing hypoglycosylation in fluids in the nervous system. So there's some really interesting stuff going on here, but it's very consistent across the patients now, and it gives us something to look for if we're going to intervene therapeutically. And ultimately what this is doing is it's enlarging the target that we can shoot at. So the more we learn about the mechanism of this disorder, or the intermediate points for the mechanism of this disorder, the better we have a chance of finding something that can intervene directly with that mechanism. So we've also, switching back to the science side, moving towards screening. So if you want to do screening in any high throughput capacity, what you really need is a good assay. And the amazing thing is so that I didn't do this. My wife actually Googled for NGLI1 assay, and what do you know? One popped up. So this is actually developed very shortly, just by coincidence, right after the disorder was discovered. And so we sent this paper to Dr. Fries and said, can you work with this? He, being an expert in glycobiology, had a very easy time adapting it and using it. And what it is, it's a really robust assay for the enzymatic activity of the missing enzyme. It shows you if you're actually getting deglycosylation activity. And so this is the kind of thing that we could run at scale through the robots in a high throughput center to actually determine whether or not there's any compound out there that makes an effect on D-glycosylation activity. So this is like firing as many arrows as you can possibly find, like say the entire library of FDA-approved compounds, all of the target, and hoping something lands somewhere in the vicinity. So it's not targeted, but there, if you fire enough arrows, eventually you'll hit something. The other thing the community's been able to help with, is to help fund the creation of model organisms where they didn't already exist. So at this point, for Engli-1, there's already available mice, flies, zebrafish, worms, and yeast. Now, I don't know if our yeast have the same wonderful nutty flavor as the ones depicted here, but I do know that we have them now. And I've also been considering the biologic route, which again I think is another route that's really important for anybody in a sort of a precision medicine odyssey when you have loss of function. And the idea here would be, can we somehow replace the missing enzyme? Can we deliver it to the cells? And in this case, we need to get it into the cytoplasm. So again, the other amazing thing about Google is that you can just Google to check to see if the enzyme already exists. And sure enough, human Engli-1 is something you can already buy on the internet. Now, the only problem here is that it's $402 for 10 micrograms. So if you extrapolate that out to therapeutic quantities, we're talking at about $2 million a week. And so our insurance company has issues with $104 million a year. But the encouraging thing here is it means it's possible. Maybe it's not cheap yet, but at least it's possible. And it turns out there's some subtle issues even here. So even if we had our hands on the enzyme in therapeutic quantities, it doesn't mean we actually have a cure ready to go. Because if you just injected this into the kids, it would actually end up in the wrong part of the cell. And so even once you start to find compounds that may be effective, you have to do some medicinal chemistry. In this case, that would involve likely pegylating the enzyme and then adding TAT peptide tags in order to increase cell penetrance. Because by itself, this particular enzyme, if injected, would go to the lysosome, and we need to have it active in the cytoplasm. So those TAT peptides would help give it cell penetrance. Peptides, which I think is pretty cool, they were basically harvested from HIV. So stealing part of a virus in order to create a medication, I think is pretty cool. The other thing I think that's doable by ulcers would be some kind of crowd screening. This is something that we've been doing first in sort of an ad hoc fashion, but which is moving forward in actually a more mature and structured fashion now. And by crowd screening, I mean just sort of turning to social media, turning to the internet in general and saying, if you had this disorder, what would you do? And really asking experts out there to chime in and give their opinion. And by doing this, we've ended up with a list of about 32 compounds, which we've been slowly working our way through, that are potentially therapeutic. And with each of the compounds, we have a sort of a proposed mechanism of harm and a proposed mechanism of action for that particular compound as to why we think it might work. So I just want to share one particular interaction that we've had recently to give you a sense of what it's like to be crowd screening on the internet. This is actually a very recent interaction. In fact, I think one of the most promising ones. So we've been interacting with a postdoc in Texas on Twitter. And his field is actually cancer, although he has interests in cystic fibrosis as well. And his particular interest is in read-through compounds. Next generation read-through compounds, if you will, a totally different approach to the small molecules that we were looking at previously. And he said, you know what, I need cells like your son's. He said, for this particular approach to read-through that I'm considering, it would be really helpful if you could supply us with some of your son's cells. Because I'm trying to target the specific premature stop that your son has. And I said, okay, well, Dr. Fries, could you ship him some cells? And he did. And about 2 weeks ago, I got an email from the postdoc which contained a blot. And it may not look like much to you, but there is, in fact, a small degree of rescued Engli-1 protein, proper Engli-1 protein in this blot, far more than we'd ever seen on any other attempted rescue to date. So even if it's only 1% to 2% rescue, that could potentially be all that we actually need to have therapeutic quantities for the patients. So it's possible, if this goes all the way through, that social media will have been instrumental not in just diagnosing the disorder, but also in actually providing a real therapy for it as well. Now, there's a lot of ifs stacked up between here and there because we have to get all the way through medicinal chemistry and stuff like that. But it's certainly promising what you can do with something like social media these days. So we've also been very fortunate that Andrew Su and his lab at Scripps has reached out and said, we'd like to see what else crowdsourcing can do as part of their citizen science initiative. And so a couple weeks ago, we launched with them the Mark to Cure initiative. So Mark to Cure is their project, and they're targeted at NGLI 1 as sort of their first big demonstration of citizen science. And this is the way for anybody, whether they're a scientist or not. So everybody in this room can go to marktocure.org and participate in this product now and measurably start advancing the work towards understanding treatment and cure. And the way Mark to Cure works is volunteers, again, they don't have to be biologists, will be presented abstracts from PubMed. And you go through a little training phase before you do this, so you get a sense of how to actually mark these things up. And volunteers start to biocurate abstracts from PubMed. And what they're doing is they're tagging it with things like diseases, and they're tagging it potential treatments, and they're also tagging gene names. So by doing this, they're making the literature accessible to deep bioinformatics. So you can sort of very, very accurately mine this biocurated data thereafter. So we've had thousands of volunteers now actually moving through thousands of abstracts to unlock the unknown known, the things that we actually knew, but that were unknown to us. And the other thing I think has been interesting to engage in is patient science. And I think it remains to be seen if this is going to be a sort of a fixture of precision medicine going forward, although I really hope it will. I hope as the barrier to science drops, that more and more patients will get directly involved. So I'll give you my own example. So I'm, as Zach mentioned, a computer scientist. My background in biology before all this stuff started was 6 weeks in high school, and it was a pretty crappy 6 weeks at that. So I didn't even know what a chromosome was when all this started. So I mean, it's been a journey. And over the past year, especially since I got tenure mostly, I've been spending an awful lot of time learning about biology. To the point where, working with some folks in medicinal chemistry and glycobiology at the University of Utah, I actually submitted a grant proposal as principal investigator to get some seed funding for developing therapeutics for NGLI1 deficiency. And so what this proposal wants to do, or what we said we would do, is actually create, among other things, a planarian model of NGLI1. And we believe this would be a reasonable model to pick because they're known to have a good seizure phenotype, which is important for our kids. They're known to show with myelination disorders, which is also important. And they have a pretty interesting secretory system. And because we have things like this absence of tears, there's a good chance that we might get some defects showing up there as well. And the other really important thing about this is that, or about planaria, is if you want more planaria, you just cut them in half. And now you have 2 planaria. So you can grow them up exponentially very quickly. I'm actually happy to say that as of last Friday, this grant proposal was funded. So starting July 1st, we're going to have some very brave worms volunteering. And people always ask, is this process, is this algorithm that we've been on, is it replicable? And the answer is absolutely yes, it is. I think this process can be repeated for other disorders because it actually already has. About a year ago, a family reached out to me and said, we'd like to do what you've done in terms of patient finding on the internet. Can you help us put together a web page? And so I just gave them some advice, how to structure a web page so that it's likely to be found by other patients. 29 days later, they had a second patient. And the interesting thing here is they had 2 potential variants of interest, and by finding another patient, they immediately went down to 1. They instantaneously knew which variant was the true cause because they could look at the pictures of these kids side by side and they knew, all right, that's it. I mean, they look like brothers. And I think what's cool about that is what that meant is that there were 2 moms out there who knew for the first time. It was really 2 moms that made this discovery who knew for the first time what this disorder really was. It wasn't the scientists, it wasn't the physicians, it was 2 moms who were the first to know because they were the ones that connected together. And other folks out there, even without talking to me, have said, well, if he can do it, so can I. And they're right. Other people are also turning to the internet to look for patients. So there's another mom out there who's set up this page and within, again, another N of 1 diagnosis, but within just a couple months had 6 patients around the world. So this is happening. And I'm very excited to be a part of, as an advisor to the coordinating center here at Harvard, the Undiagnosed Diseases Network. So for the past 9 months, I've been working with people like Zach, Rachel Ramoni, and others to figure out, can we scale this process up to every new diagnosis that comes through this network? And I'm confident that we'll actually be able to do this. And I've also been sort of expanding on how you go about using this process. And I think one of the other things you can do is to use Wikipedia. So this is a point that I think is vastly underappreciated in both the patient population and in the medical community. So I'm telling you here today that I think Wikipedia is our secret weapon for finding patients. So what is Wikipedia? Well, in case you've been in cryostasis since the late 90s, we have a lot to catch up on. But Wikipedia has become the world's database. I mean, anybody can view Wikipedia, anybody can edit Wikipedia, and it's what patients and parents search. I mean, every patient ends up with an MD by Wikipedia if they go at this long enough. And the other really critical factor here is that Wikipedia is a very authoritative source, which means it tends to rank very highly in Google results. So you can sort of skip right to the top of Google search engines, Google search results for patients and parents if you modify the entry to somehow get back to you. So to give you a sense of how to do this, and actually a lot of other tips, I've written a blog post on how to do rare disease matchmaking over the internet. And I've compiled my list of tips here. And one important part of this post is actually a little interactive web app that will help you generate your own Wikipedia entry. So you can fill out the information for your particular disorder, and it will generate the text that you need to put on Wikipedia. I would actually have it automatically upload for you, but that definitely violates Wikipedia's guidelines. And then, once it's up there, you now have this sort of reverse Google Dragnet operating to help bring the patients to you. And if you, for some reason, don't want to edit Wikipedia, or in addition, you can also just buy AdWords. So AdWords are incredibly cheap. And this is not asbestos litigation here. This is like rare, bizarre gene names, which are probably about 5 cents a click. So it does not cost a whole lot to buy AdWords. And then you go straight to the top of the search results right away. So there's you don't have to have a blog in order to do something like this. You can cheat. There's ways to cheat and get right to the top for the stuff you care about. So to sort of wrap this all up, let me sort of go back over the algorithm for N of 1 medicine. And what I'll do is I'll present it to you as a flowchart, and I'll trace out some parts of it that we've been through. And what I think of when I think of precision medicine and this algorithm for it, I'm thinking of here is sort of next generation genetic counseling. I think this is the kind of thing that genetic counselors in the future will help patients traverse. So it's a process that starts with being undiagnosed and then moves to sequencing. And then once you've sequenced, I think you can split apart and in parallel turn to the web, turn to Wikipedia, turn to gene experts, and potentially even functional studies right off the bat. Because functional studies are expensive, but if you can find that one of your variants matches and that phenotype also matches just by looking on the internet, that's much cheaper. So I think people should turn to the internet much sooner than they might think otherwise. So even if you don't have even if you have several variants, I would still put them up there and say that these are plausible diagnoses. Once you've got all that data, hopefully you have an interpretation. And once you've interpreted the data, you're going to get roughly 4 things that might happen next. So the 4 things that could happen are, for the mutation, well, you might not get an answer at all, but hopefully what you find is that you've either got a gain of function, partial loss of function, total loss of function, or a change of function, or perhaps a change of structure if it's a structural protein like dystrophin or something like that. And there's going to be a whole network of things you can do behind those. But in tandem, regardless of which of these it is, you can also engage in crowd screening. So you can say, well, we've just been told that this is what we have. What do you experts on social media, Twitter, Facebook, and Reddit think we should do? And if you can start rounding up patients in the internet, you can start doing that deep phenotyping. You can do that natural history, which yields so much insight into the disorder. So let me trace out a little bit on total loss of function for just a second, because this is the path I've been on, and there's a lot you can do even with the total loss of function. So and some of this applies to the other paths as well. You can do model organisms, you can do structural analysis, you can do enzyme synthesis, you can consider metabolic diets, and you can even do cell biology to do assay development. And all of these in turn lead to even more things that you can do. So for example, if you've got a good model organism, you can consider doing a suppressor screen. So and if a good target turns up on a suppressor screen, if you find a gene that when suppressed actually improves the phenotype, you go from having to do enzyme replacement to doing an inhibitor, which is closer to what pharmaceutical companies actually have been historically better at doing. I won't go over all the parts of this, but there's a lot you can do in the event of even total loss of function. Like for example, if you do structural analysis of the protein, of the mutant protein, and find that it might have some residual activity, like perhaps if the sort of the key catalytic domain is preserved, then you might try upregulating that mutant behavior or upregulating the production of the mutant to see if you can boost that latent activity. just to focus on one in a little bit of detail, because I think this is sort of a critical low-hanging fruit, I want to talk about metabolic diets. Because sometimes, there's something you can do right off the bat that doesn't require doing a whole lot. And so for a metabolic diet, you just want to consider the logic of the pathway. So let's just look at it very abstractly for the moment. Let's just say that, it's a pathway that turns an A into a B. And there could be multiple A's and multiple B's here, but pathways turn this into that. They're functions from a computer science perspective. When a pathway breaks, here's what happens. Presumably what's happening is that you've got perhaps too much A and not enough B. And it could be that both of these are actually contributing to the mechanism of the disorder. But this gives you a hint as to how you might alter diet in response, because you can start to ask, can you restrict A and can you give B? Are there ways to do this? And there are other genetic disorders where this actually is more or less a completely effective treatment. An example of PKU, you want to restrict phenylalanine. And if you do, it's more or less a perfectly effective treatment for the disorder. For the CDG, which is phosphomannan deficiency, if you can provide mannose, it is effectively a complete treatment for the disorder. So this obviously is going to work for every disorder, but for some fraction of disorders, this might provide some very immediate relief if you're willing to try it. And of course, you might ask, well, what happens in the case of NGLY1? So we've considered this logic too, and it's been interesting, very interesting, because in just the past 3 years there have been some new insights into the mechanism of disorder. We have a much better sense of what's going on in the cells of these patients now. So we used to have this view that it was a certain kind of misfolded glycoprotein piling up. Well, now we have a much more accurate view of what those misfolded glycoproteins look like. We know that they actually have a particular sugar, a particular monosaccharide on them, and in fact that this monosaccharide is what allows the proteins to actually aggregate together. So we have these protein aggregates sitting in kids' cells, and it's predictably locking up this monosaccharide. So we thought, well, if we've got a deficit of this particular monosaccharide represented by the blue square, maybe we should be supplementing with that. And there's actually been lots of other evidence coming out over the past year that this really is a part of the disorder, including dysregulation of the free oligosaccharides in the cell. And the interesting thing about this monosaccharide is that when I Googled it, I found out that it was actually available on Amazon for about 25 cents a day. So I thought, and why not? So I ordered it. I took a bottle myself, because in my household I am the FDA. And when it didn't kill me, I went ahead and gave a little bit to my son. And the amazing thing is that 3 days after we gave it to him, he cried his very first tear. So this was. And I actually have a picture of all of his tears because he's cried many more since then. And I did what any parent would do. We collected the tear, put it on dry ice, and shipped it to a lab in California for analysis. So this may be one small tear for NGLY1, but I think it's really represented sort of an ocean of science for the patient community. And the other interesting thing that happened, this didn't happen immediately, this happened very subtly over about a 3-month period. We also noticed that his nighttime seizures stopped. So he used to have a seizure so predictably at the same time every night that we literally could set an alarm and be there for it. In fact, when we took him to NIH the first time, I told the technicians on his overnight EEG exactly when it would happen, and I was only off by like 10 minutes, which is not bad considering that we were 2 hours apart in terms of time zones. So these things were, every night, literally every night, we were waking up to these seizures, and there was nothing we could do to stop them, but those disappeared after about 3 months on the sugar. So presumably that's because we've corrected some fundamental mechanism here. Now, I wouldn't say he's cured by any means. This is still a pretty horrific disorder, and there's a lot of developmental damage that's been done, but he has a much higher quality of life today than he used to. So let me trace out some other paths in brief on the precision medicine flowchart. So if you have a gain of function, there's some other things you can do right off the bat. And I've looked at other patients to see whether these things are realistic too. So you can look at RNA downregulation. You can look at inhibitors. You can also consider things like model organisms or even a metabolic diet here as well. And I just want to share one story from a patient in California. I met this girl who was diagnosed through genomic sequencing several years ago to have a de novo gain of function in a particular gene. There's some very thoughtful functional analysis done by some very dedicated scientists to show that really was gain of function. And she'd been waiting for 2 years for a next step. And what I did is I just typed gene X inhibitor into Google. That's all I did. And sure enough, something popped up. And this is what killed me. It's like, anybody who'd been doing those functional studies surely knew that if you have a gain of function, the right next step was to look for a suppressor or inhibitor of some kind. Any of them could have typed this into Google. That family didn't have to wait 2 years to take the next step on their journey, which at this point is medicinal chemistry, turning this compound into something that's safe and reasonably bioavailable for their daughter. So one piece of advice I would have for anybody practicing precision medicine is that make sure that when a patient leaves your office that you've told them what the next step is, because it may be obvious to you, but it may not be obvious to them. In the case of partial loss of function, you can do a lot of the same stuff that you do for total loss of function. I won't labor that point. And then for a change of function, you pretty much want to do everything that ends in omics. So if it's got omics on it, if it's change of function, change in structure, you're in omics land at that point. Because I think it's probably the most challenging case to deal with next to loss of function. And I think the step that anybody can start with on this flow chart, the step that you don't have to even have a diagnosis to do, is to actually advocate. So to talk to doctors, talk to politicians, and start pushing for the regulatory and legal changes that need to be in place to make precision medicine a reality, because there's a lot of roadblocks in the way. There's a lot of scientific roadblocks, but there's also a lot of artificial roadblocks as well. So I'll share one story from back home in Utah of my wife and son. So they actually went to the state house and the state senate this past year, and they testified on behalf of the Right to Try bill. What the Right to Try bill does is it gives any Utah resident with a fatal disease the right to try anything past phase 1 safety testing with the FDA. And the bill passed unanimously in both houses, and when the governor signed it, he invited my wife and son up there for the bill signing and even gave them a signed copy of the bill. So my son is very excited that this bill is now law, or will be law as of July 1st in Utah. And I think this is the kind of stuff we're going to have to do if precision medicine is going to become a reality. This is just an opening salvo on the regulatory changes that have to happen. And then in terms of running the algorithm at a systematic scale as opposed to a one-off basis, I think that's a really big challenge. But I'm very excited that next year is my sabbatical. And as a matter of fact, it starts next week. And I'm moving here to Boston for my sabbatical. And I'll actually be, thanks to Zach and Rachel Ramoni, right here in the now Department of Biomedical Informatics, working with their amazing team to try to figure out how do we scale this up. And again, this is under the auspices of the Undiagnosed Diseases Network, because everybody who comes through a program like that, everybody who gets a diagnosis or even a near diagnosis through a program like that is going to want to follow a path like this. And we have to figure out how to scale this up. So, parting thought. If there's, again, one thing I could impress upon you today, if there's one thing you leave here with, it's that the phrase "not actionable" doesn't belong in our medical vocabulary anymore. Because in the absence of any other alternative, you can always do science. Thank you.